bank interbank charge kay video magyap ini na sa niya. O sige ka do. Padarak ng tanira pa mas Davao. Dari ako ka anak brother. Tagain na dito Arlene. O to siya tawagan ko kagabi Arlene. Ginaw na diudugay ng istorya imbis na magbasa pagkadi. Taman. Pero na. Tapos mo kaya nagpata na lang sa iyo. Okay na siya? Ako na, nagsige pa po chika. Jesus Christ. Good morning.
Good morning, Doc.
Good morning po, Doc.
Okay. So we'll continue with the lecture. So today we're going to take up the next chapter. So after the blood, RBC. So now we'll be going to discuss the WBC. So it's resistance of body to infection 1, because the next chapter after this resistance of the body to infection 2, that's on immunity. So in this chapter we will be discussing only the different cells in the white blood cells, their functions, their origin, where they came from, how they came to be. And then the next chapter will be on immunology. Okay. So ini siya ini pares lang. Okay. So this slide, this will show us a picture of a macrophage, how it looks like under an electron microscope. So it has pseudopodia, like tentacles trying to attract an invading bacteria. So the yellow ones, if it's yellow in your screen, this represents bacteria, bacilli in particular. Okay. So next, siya, wag man lagi mag-move siya. Wait, excuse me. Hindi ko na bubugya ini pag-aga. Okay na. Kailangan mo kung magpindot. Ito pindot mo. Sige na daw. Okay. Okay na. Second slide na to. Okay. So, defense mechanism of our body against infection. So, later on, we will learn that there are two types of defense. The natural or the innate defense system, and then the specific system, that's the immune system. Okay, so the defense system of the body involves mainly the WBC and tissue cells derived from the WBC or leukocytes. Okay, so we will later on learn what are these WBCs and then these derived cells. So how do they prevent disease? They prevent disease by actually destroying invading bacteria or viruses by way of phagocytosis. Okay. So again, the picture on the right side of the screen depicts a phagocytic cell, the blue cell, trying to engulf, highlight marker pointer ni siya, trying to engulf bacilli. These are bacteria being engulfed by the bacilli, and it goes into the cytoplasm of the bacterial cell, sorry, the macrophage or the phagocytic cell. Okay. And then this is non-specific defense. Well, specific defense involves the production of antibodies. And these are the red one, the Y-shaped ones are the immunoglobulins, and the yellow round irregular shape round yellow thing represents the antigen. So you see the antigen is being attached to an immunoglobulin molecule. So the attachment, if you take note, is only at this portion, no? Not at the tail end. So this is the variable portion for where the antigens are attached. So this is just a representation of the specific defense, and the other one is, this one is the non-specific. Ito iniya nga arrow siya. Kini nga ganang cursor na nakikita ni student ano kini so pwede na mo himo niya pointer ano pero mas magkano na red ano first kulo po okay so okay so they actually prevent disease by destroying the bacteria or viruses by phagocytosis, or another method is by formation of antibodies and the sensitized lymphocyte that promotes activation or destruction of the invading organism. Okay, so next slide is a slide on the type of WBC. They are divided into two major groups: those with granules and those without the granules. Okay, granulocytes and the agranulocytes. You learned this in your histology class. So the granulocyte, those with granules are your neutrophils, eosinophils, and the basophils. And those without the granules, you have the monocytes, the lymphocytes, and then these are a specialized type of lymphocyte, the plasma cell, which is the precursor of the antibodies. Okay, so these are the so-called member of the search and destroy group. So again, the normal blood count, WBC count, is between 5,000 to 10,000 per microliter of blood. And, this one, everybody should know, we should know, because how can we tell if it is not abnormal if you don't know the normal? Okay, so you're given a CBC result with a WBC count of 20,000 per ml of blood, so it's no longer normal. So there must be something. The patient must be having some form of infection. Okay, then you look down at the breakdown of the WBC. So you have difference. We call that differential count. So neutrophils 62%. That's the normal. So you see 62% neutrophil normal. Then eosinophils, di man lagitin yan na ano siya. This one. Di ba nga mga picture hit estudyante? Di mo nakikita. Then basophils, 0.4%, monocyte 5.3, and 30% lymphocytes. This morning, just coming to school, my nephew texted me and said, "Nanay, this is my CBC. Okay, look, oh, you have a very high neutrophilic count, low lymphocytic count, because he was having fever, cough, and he went to Mandawi Center for a nasal swab for COVID, and it turns out negative, taas and neutrophil. So I said, you must be having a bacterial type of infection. So just continue, finish that antimicrobial for a week, and then mag-rest ka muna. Do not go out. Stay at home, even if it's not COVID, because your resistance at this point is low. Then you might catch COVID outside. So this is how they came about, where the origin. So if you remember yesterday, that from a pluripotent hematopoietic stem cell, the cells, that plurihematopoietic stem cell will differentiate into myeloblast and the lymphoblast, and then from the myeloblast series, you will have the promyelocyte and then the megakaryocyte. So the promyelocyte is the cell origin of the granulocytes. Okay, your neutrophils, the eosinophils, and the basophils. And then again, from the same lineage, same family, myeloblast series, aside from the promyelocyte, you will also have the monocyte. No? So you have an immature monocyte, neutrophilic monocyte, young monocyte, and then these are the ones at the bottom of the graph are the mature. So you have the neutrophils, these are the monocyte. Then the megakaryocyte are the origin of the platelet. Okay, So your myeloblast gives rise to myelocytes. And then your lymphoblast, on the other hand, on the other end of the spectrum, you have the lymphoblast, which will give rise to the lymphocytes. Then you have three populations of lymphocytes: the T lymphocytes, the B lymphocytes, and the NK. NK, or the natural killer cells. Okay? So upon antigenic stimulation, you activate your B lymphocyte. It will differentiate into a plasma cell, and the plasma cell now will form the antibody molecule, the immunoglobulin molecule. So megakaryocyte, they are found in the marrow. They become blood platelets after fragmentation, and they are involved in blood clotting or hemostasis. So again, normal count of platelet, so it's a 300 per microliter. But yesterday, if you remember the slide I showed you, it's from 250 to 400. So within that range. And then it has a lifespan of 10 days, a short lifespan of platelet. That's why in dengue, the rationale in dengue, why we request for a fresh blood, not the one that has been in the blood bank for several days or weeks, is because of the platelets. In dengue, there's bleeding, you need platelet, right? For blood clotting, for hemostasis. And the blood in the blood bank has been there maybe for several days, so wala na ang platelet. So nangamatay na si platelet. But, so we need a new fresh blood because the platelet are still active. That's the rationale. Okay. So granulocytes and monocytes are formed in the bone marrow, while the lymphocytes and the plasma cells, they're formed mainly in the various lymphogenous tissues. So what are these? What are the examples of lymphogenous tissues? The lymph glands, the spleen, thymus, the tonsils, the Peyer's patches in the intestine, and then in the bone marrow. So they are mostly stored in the various lymphoid tissues, and a small fraction is continuously circulating in the blood and in the lymph. So from the blood to the lymph and back to the blood. So, vice versa, kumbaga, sasakyanan. Okay. So most of them are stored in these lymphoid tissues, and then a few of them are in circulation. So they are being released into circulation in case of there is an infection going on. So the granulocytes, 75% of them are stored in the bone marrow. So the eosinophils, basophils, neutrophils. So they're in the bone marrow because it says in the barracks, because they are at the undergrad, or in high school in our science class. We remember our science teacher telling us that the neutrophils are the policeman in the body. No, they are the first one to be at the scene of a crime. So yun. And the rest are doon lang sa detachment, stored in the barracks, and 25 of them are on patrol or in circulation. So the one in circulation, it's a 6-day supply. So sa pasimana ng patrolya. So the lifespan in the blood, they last for 4 to 8 hours. In the tissue, longer, 4 to 5 days. But in the case of an extreme severe infection, the lifespan of the granulocyte is shortened. While for the monocyte, they stay in the blood for 10 to 20 hours. And then when they migrate to the tissue, they are no longer called monocytes. They become macrophages. So they enlarge and become tissue macrophages with a lifespan that is much, much longer. So they can stay in the tissue for several months unless they are killed in action. So, okay. So your neutrophils and your tissue macrophages, they attack and destroy invading bacteria, viruses, and other injurious agents. Okay, so neutrophils will act on the tissues and in the blood, while macrophages are only in the tissue, di ba? Because in the blood, they are known as monocytes. Okay, so how do this enter the WBC or white blood cell enter the tissue spaces? We do it by a process known as diapedesis. Okay, so in the endothelial lining of the blood vessels, there are minute pores or holes wherein the WBC, by way of an amoeboid motion, they leaked out of that endothelial opening, small opening, and they are attracted towards the infection site. And they are attracted towards the site of infection by a process known as chemotactic or chemotaxis. Okay, so the inflammatory reaction will release chemotactic substances which will attract the WBC towards the site of inflammation. So again, this is a scheme to schematic kind of representation of how we defend, how the body defend against body defense against infection by phagocytes. So okay, so start with you have an invading antigen or pathogen contact with the phagocytes. So what will happen? The phagocytic cell, your macrophage or the other phagocytic cell like neutrophils, will try to engulf that by way of phagocytosis. So inside the phagocytic cell, it can lead to an intracellular killing of the microbe. Then the other method is if the phagocyte is unable to kill that invading microbe, so it also secretes chemicals, okay, secretion of chemicals by the phagocytes, which will lead to regulation of the inflammatory process or to extracellular killing of the microbes or activation of the clotting and the anti-clotting pathways, and then hormonal regulation of overall bodily processes to the infection. Okay, so this is a schematic representation of phagocytosis. So when an organism, a microbe, so this rod-shaped blue thing represents a microorganism. It is when it's taken up by a phagocytic cell. So let's say this one is a macrophage. By way of phagocytosis, so it's engulfed by the phagocytic cell and it goes inside the cytoplasm of the phagocytic cell. So in the cytoplasm, they have a phagosome. So it forms a phagosome. The bacteria or the microorganism is engulfed by this phagosome. So to form into parang bacule. Okay. Yes. Excuse me. Then inside the macrophage, you also have this lysosome. It's a bacule filled with enzymes. It contains those elements that will neutralize or will kill the organism. So the phagosome fuses with the lysosome to form into a phagous lysosome, and it starts the intracellular killing of that microorganism. So what happens, the microorganism, if you notice from a solid rod-shaped structure, it becomes fragmented, okay, by the action of the enzymes found in your lysosomes. And then it will be released into the cell or outside of the cell. Okay, so release of end products into or outside the cell. Okay, so neutrophils are able to phagocytize 3 to 20 bacteria, and they have proteolytic enzymes. While macrophages, since they are bigger than the neutrophils, they can phagocytize as much as 100. And they have proteolytic enzymes and lipases. So lipases are enzymes that breaks down the lipid structure in the bacterial cell wall. So both of them, the neutrophils and the macrophages, can also kill bacteria by using oxidizing agents. So they produce this oxidizing agent. You have your superoxide, your hydrogen peroxide, and hydroxyl ions. Okay. So these are just the macrophages in the reticuloendothelial system. So in histology, you learn that as these macrophages in the different areas, different organs, different structures in the body. Some of them have a particular name, like the copper cells. These are the macrophages lining the liver sinusoids in the liver. And then histiocytes, these are found on the skin. And then aside from the skin, you also have this in the lungs, in the other organs and tissues of the body. So then alveolar macrophages in the alveoli of the lungs, in the lymph nodes, on the spleen, again in the liver, that's copper cells, and then in the bone marrow. Okay. So sequence of events in non-specific local inflammatory response. The first is an entry of bacteria into the tissue, and it causes tissue injury. And then this tissue injury releases chemicals to initiate the succeeding events. So the first event is you have vasodilation of the microcirculation in the infected area. So leading to an increased blood flow. So initially, di ba, if you have an infection, medyo mainit-init. Signs of inflammation, di ba? Increased because of increased blood flow, so a little heat is felt, and then swelling. Large increase in protein permeability to the capillaries and the venules in the infected area. So it will lead to diffusion of protein and filtration of fluid into the interstitial fluid. So yun, in inflammation. So there will be swelling, redness, swelling. Then chemotaxis, the next event, is movement of the leukocyte from the venules into the interstitial fluid of the infected area. Then the next step is destruction of the bacteria in the tissue, either through phagocytosis or by other mechanism. And the last event is tissue repair. So this is uh, you know, a diagram of an activated macrophage releasing these cytokines, tumor necrosis factor, interleukin-1. This will stimulate your endothelial cells, fibroblasts, and lymphocytes, releasing, remember what GM? GM-CSF, granulocyte, monocyte, colony stimulating factor, granulocyte, colony stimulating factor, monocyte, colony stimulating factor, stimulating the bone marrow to produce more granulocytes, monocytes, and macrophages. Okay, so what are cytokines? These are broad and loose category of small proteins that are important in cell signaling. Okay. So a lot of cells will produce cytokines. Cytokines are peptides, and they cannot cross the lipid bilayer of the cell to enter the plant cytoplasm. And then they have been shown to be involved in autocrine, paracrine, and endocrine signaling as immunomodulating agent. I think in histology, you have learned what an autocrine, paracrine, and endocrine. No? So I remember this. I once also taught histology at DR. Nagadinikuan na pusing siya picture tadi. May ganyan ituan. Mga sa kita doon, diba? Pwede ba guti-guti yun niya para damo makita? Orange uniform. with Rosa.
Hindi, tulog. Hindi ko anong ganina. Kayo ni pharmacy yan.
Ano niya may tugman?
Ano lang yun. Ate kasi hindi mo lang Tuesday pa kasi. So, ano lang ko.
Hayaan mo.
Opo.
Sayang talaga naman. Okay, so we'll just continue. Okay, so yan, a cytokine may affect in three ways. Autocrine, by paracrine, or an endocrine way. So autocrine, your cytokine binds to a receptor on the cell that secreted, that produces it. Okay, so that's a cytokine. While a paracrine, the cytokine binds to a receptor on a nearby cell. So this is the cell that produces cytokine, so it binds on a nearby cell. While endocrine, it binds to cells in the distant part of the body, so that cytokine is being transported to the distant part of from the cell that produces it through blood circulation into the other parts of the body. That's how the cytokine acts. So you will get to read or hear about cytokine in COVID. There's such a thing as a cytokine storm na sobrahan ang action sa cytokine. Okay. So again, this graph shows the different cytokines and then their target cells and then their function. Medyo, I understand medyo at this point, medyo ano siya, ano ba tawag niyan? Medyo ma-overwhelmed ka of this one. There are a lot of this, but we only just listed down a few. I remember when I went for the postgrad, kaya damo yan, iniya, damo ngaran, damo ng mga IL, etc. Okay? But we just listed down some of the few things, important cytokines we need to know. So interleukin-1, usually it's abbreviated as IL, okay? Then the tumor necrosis factor or TNF, and then interleukin-6, so I lump it into one group. So their target cells are the helper T cells, certain brain cells, and numerous systemic cells. So in COVID, you have these three. They stimulate, what is the function of interleukin? Interleukin-1 stimulates interleukin-2 secretion and interleukin-2 receptor expression. And then it causes induction of fever, induced fever. And then they stimulate systemic response to inflammation, infection, and injury. So this group, interleukin-1, TNF, interleukin-6. While the interleukin-2, the target cells are your helper T, the cytotoxic cells, your natural killer, and your B cells. So the function is stimulate proliferation, and it promotes conversion to plasma cells. So conversion of the B cells, because this is the only cell that can be converted to a plasma, the B cells, B lymphocytes. Then interferons, these are also cytokines. So most cell types are the target of interferon, so part of a non-specific defense. It stimulates cells to produce antiviral proteins, and it's a non-specific response. Then we have interferon gamma. The target cells are the natural killer and macrophages, and it stimulates proliferation, secretion of cytotoxic compounds. If you read further, you will learn that there are several subpopulations of interferon, not only gamma. Okay. Then chemokines, again, these are cytokines. The target cells are your neutrophils and other leukocytes. So what does it do? It facilitates accumulation of leukocyte at the site of endothelial cells, leukocyte injury and inflammation. So these are the substances that will attract your neutrophils and other leukocytes towards an area of inflammation. Then CSF, or the colony stimulating factors, these are also cytokines. Their target cells are the cells in the bone marrow. They stimulate the bone marrow to produce a particular cell, produce a monocyte, a granulocyte, okay, a lymphocyte. So stimulate proliferation of neutrophils and other monocytes. Okay. So this is just, I just added this 2 years ago. Mechanism of fever by interleukin 1 and tumor necrosis factor. So in the case of infection, so we have an exogenous pyrogen. Then your endogenous pyrogen, interleukin 1 and tumor necrosis factor. What does it do? It increases enzymes of the cyclooxygenase pathway of the arachidonic acid metabolism. So this one will send signal to the hypothalamus, increased prostaglandin E2. PG is prostaglandin, which will stimulate neurotransmitter production and reset the temperature set point at an elevated level. So the temperature set point, it's found in the hypothalamus. Okay. Okay, so this one is this one is easier to understand than the ontogeny of the WBC. So from the hematopoietic stem cell, from the blood stem cell, you have the myeloid series and the lymphoid series, right? So from the myeloid stem cell, it gives rise to RBC, okay, and then to the myeloblast. So the myeloblast gives rise to this, granulocytic cells, the eosinophil, basophils, neutrophils. And from the same myeloid stem cell, again, it will differentiate depending on the stimulating factor and differentiation factor. Remember yesterday, there was a cell differentiation factor, growth stimulating factor. So cell differentiation factor stimulates this one to become megakaryocyte, or the platelet. Then on the other hand, the lymphoid stem cell comes from another. It does not come from the myeloid. So the lymphocytes are from the lymphoid stem cell. So the initial cell is a lymphoblast, and then the lymphoblast differentiates and become a B lymphocyte, a T lymphocyte, and a natural killer cells. So there are three product of the lymphoblast, the B, the T, and the NK cells. So these are all, ito sila mga WBC, the granulocytes, and then this one is the agranulocyte, the lymphocytes. Okay. So this is just a summary of the various WBC. Their site where they're produced, and then their function. So we'll just run through it fast, because again in the next chapter at the start, okay, so neutrophils they are all, if you notice, they are produced in the bone marrow. Neutrophils, basophil, eosinophil, monocytes. Okay, so the function of the neutrophil is phagocytosis, so it's one of the phagocytic cell, and then it will release chemicals involved in inflammation that can cause vasodilation and chemotaxis. Then the basophils in the bone marrow, they carry out functions in the blood similar to those of the mast cells in the tissues. And then eosinophils destroy multicellular parasites, so that in case of parasitic infection or in allergy, this one increases in value. Okay, so you have to learn the normal values to know that you are dealing now with an abnormality or with a disease or an infection. Okay, your monocyte carry out function in the blood similar to those of macrophage. because they are the precursor of the macrophage. So the monocyte, when it goes into the tissue, it becomes a macrophage. Then, yeah, the tissues are transformed. Okay, more. The lymphos, again, they mature in the bone marrow, so they become destined to become the B cells and the NK cells. And those that mature in the thymus, they become the T cells. That's why they're called T, thymus, B, bone marrow. So in the textbook, you may have heard of the bursa of Fabricius in chicken. That was the first time they observed that the B cells, but we are not chicken, we do not have a bursa. What we have is a bone marrow. Okay. So B cells and NK cells are from the bone marrow, right? And then the T cells matures in the thymus. Then they are activated in the peripheral lymphoid organs. So in the bone marrow or in the thymus, they transform or differentiate regardless of, independent of antigenic stimulation. So they serve as recognition cells and specific immune responses and are essential for all aspects of this response. Okay, so the population of lymphocyte, the B cells, they initiate antibody-mediated immune responses by binding specific antigen to the B cells plasma membrane receptors, which are immunoglobulins. Then during activation, they are transformed into plasma cells, which secretes antibodies. And then another function is they act as an antigen presenting cell. So present antigen to a helper T cells. Then the other population of lymphocyte, you have your cytotoxic and the helper T. So cytotoxic T cells are also known as CD8 cells. So what does CD stands for? Miss Calvero. Kamatadi did it screen. Are you around, Miss Calvero? Magsinta na siya. Manatingog. Do you know what CD8 stands for? Compact disc? Or ano ba?
Cluster differentiation.
Cluster of differentiation.
What cluster of differentiation? Okay. So your cytotoxic T cells or CD8 and then the helper T are CD4. Actually, there are a lot of subset. Again, when I went for the postgrad in micro, nagsuka-suka ko ani sa kadagho nga CD na ani CD25, CD8, CD4, may CD. Kadamo nga CD. But sa katapos-tapusan inilahara nga duhaan ano. Kumbaga ang iba mga ano na lang tira. Pampagulo sa utak ko. Because we have a professor at the who was discussing immunology, British from UK, import nga, lecturer. You're trying to impress us with all the CD. Sa notebook, sa ka-page, puro CD. Okay, so CD8 or your cytotoxic T, anong function niya? It binds to antigen on plasma membrane of the target cells. So what are these target cells? Virus-infected cell, cancer cells, or tissue transplant, and directly destroy that cell. So itong mga transplant rejection, tungkol dito cytotoxic. Then you'll have helper. Helper T, they secrete cytokines that help activate B cells. And they also activate cytotoxic T, or NK, and macrophages. Helper nga, damo yan, mabinuligon. Then another population of lymphocyte, the NK, this is not part of the specific immune defense, but non-specific. It binds directly and non-specifically to virus-infected cells and cancer cells and try to kill them if kaya niya. Then they function as killer cells in antibody-dependent cellular cytotoxicity. So Discuss ito in the next chapter. Then plasma cells, these are B cells that are produced in the peripheral lymphoid organs. That a B cell that differentiate into a plasma cell upon antigenic stimulation. So if there is an antigen coming into the body, it will stimulate the B cells, and then the B cells are transformed into a plasma cells. And these plasma cells are the ones that will produce the antibodies. Okay. Then another cell in this group are the macrophages, produced in the bone marrow. Then they reside in almost all tissues and organs of the body, and their origin are the monocytes. So they differentiate from the monocytes. So the function of this is, the main function is for phagocytosis, and then they are involved in extracellular killing by a secretion of toxic chemicals, then the process and present antigen to a helper T cells. So what the macrophage cannot kill or cannot contain or inactivate, it can help kill that antigen by processing it and then present that to a helper T. Then it secretes cytokines involved in inflammation, activation, and differentiation of helper T cells, and also in systemic response to infection. Okay. So, tapusan na din. Dendritic cells, these are macrophage-like cells, but they are much, much larger, and they are capable of phagocytizing a lot more than the macrophages. So they are produced in almost all tissues and organs of the body. And one is in the microglia of the brain, of your CNS. Their function is same as macrophage, but their phagocytic capacity is much, much bigger than the macrophage. We said in that slide on the phagocytosis that the neutrophil can engulf 20, while the macrophage can engulf as much as 100, and this one more. Sobra 100 siya. Then the mast cells from the bone marrow, they reside in almost all tissues and organs. So they are they differentiate from bone marrow cells. What is the function of the mast cells? So they produce substances like histamine, so release histamine and other chemicals like heparin involved in inflammation. So release histamine and other chemicals involved in inflammation. So that, I think that ends the chapter. So we'll go to the next, and ito siya, paano to, how? Ha? Magabay. Thank you. Yes.